Hello, this is Interactive Biology and my name is Leslie Samuels. Today we're going to go over the anatomy of a neuron, and as well as the physiology of a neuron. This collectively is known as a neuron, which we're going to find in your peripheral nervous system. First we'll start from the left and move on to the right. These orange fiber-like structures that are projecting out of this big circle are known as your dendrites. They receive electrical information and bring it into the cell body. Now this black big circle is known as your cell body, or also soma. And this smaller blue circle within that is known as the nucleus of your neuron. Now, as the electrical information is going through your dendrites into your soma, it's going closer and your soma, and then this long tube, which is collectively known as your axon. The blue rectangles within these tubes are known as your Schwann cells. Now the Schwann cells in the peripheral nervous system make up the myelin sheath, which its basic function is to make the action potentials go through this axon much faster. In between these Schwann cells you have something called the nodes of Ranvier, which are very important to work together in conjunction with these Schwann cells to perform saltatory conduction. As your electrical signal reaches the end, known as your axon terminal, things such as your neurotransmitters will be released and will go and connect to over a synapse to another dendrites of a neuron. And that's the basic anatomy. That's the basic anatomy of a neuron. Now this is part 2, which is the physiology of this neuron and how an action potential works. First, we'll be looking at the different parts of action potential. We'll first be looking at when graded potentials reach threshold, depolarization, repolarization, hyperpolarization, and then reaching back to resting membrane potential. First, this neuron as follows is at resting membrane potential. Now, as you see in this chart, there's many things called graded potentials. Now, these graded potentials are when these neurons are being stimulated, however, they're not reaching the threshold to form a full action potential. However, when a graded potential does reach threshold, the following steps will occur. First, we'll look into depolarization on this big axon. First, on this big neuron. In resting membrane potential, inside of your axon, you have many potassium ions, and on the outside, you have many sodium ions. Now, when the stimulus reaches the axon hillock, slowly voltage-gated sodium-potassium channels open, and your sodium starts to go into your axon with the potassium already there to slowly increase the charge within your axon. Now, as your axon in the beginning starts to gain a much more positive charge, it starts opening up more voltage-gated cells, which helps the sodium enter throughout the axon. Now, this is why, when you refer back to this chart, depolarization is when you're increasing the charge within your axon. Therefore, it's becoming more positive. Now, it will reach about 40 millivolts positive. However, the equilibrium for your sodium within your axon is going to be at a charge of 58 millivolts. That's what it wants to reach. So when it reaches 58 millivolts positive, can you look at the neuron again? That's when your voltage-gated channels will open once again, and this is where your repolarization starts. Now, in your repolarization, at this point we've reached our, because our sodium wants to reach its equilibrium at 58 millivolts, we've gone up to 40 millivolts, and at this charge, the sodium-potassium channels open up once again. Now, due to the large amounts of positive potassium and positive sodium ions within the axon, they're going to start repelling each other. So due to this, the potassium is going to start leaving the axon. So you're going to have positive charges that are leaving the axon, and this is what you call repolarization. However, However, the equilibrium for potassium is negative 93 millivolts, which is less than the negative 70 resting potential. Now, that means that so much potassium is going to leave that the charge is going to go below its resting membrane potential, and that is known as hyperpolarization. However, once it reaches your negative 93 millivolts, your sodium-potassium channel is open, and what happens in order to reset it at resting membrane potential, you're going to have 3 sodiums that leave, 2 potassiums that come back. 3 sodiums that leave, 2 potassiums that come back, 3 sodiums leave, and just so on, back till its at potential is known as your refractory period. And this is our resting membrane potential, and is able to go through another stimulus. And this is how action potential in a neuron works.